So welcome to the review session for Exam 3. Um, I will open it up to questions first. Brian, I see that you have joined us, but I don't see that you have any audio. So can you hear me, Brian? You might need to chat in order to, let's see, let me chat with you. In order to tell me that if you can hear or not. Okay. And while we're waiting for Brian, we can go ahead and start if Paula or Mike, if either of you have any questions to start off with. Otherwise, I'd be happy to start going through the review starting with the
I
study
do.
guide. Okay, yep.
I know the guide says to focus more on 6 and 13.
6 and 13, yes, that's true. Less questions, maybe 10% or so, 10-12% will be from chapter 7, so definitely know chapters 6 and 13 in more depth. You can kind of get a sense of that from like the questions that I have for the open response as well as the detailed questions down below.
Right. So then I take it, details of, the pathways and cycles. We don't need to go that deep. I didn't see anything about it really on the guide except for a little bit about the fermentation and I guess the very basics of, catabolism and stuff, but that seems about it.
Right. So for chapter 7, and I'm going to share the screen with you now so that I can go over to, let's see, how do I get this to get smaller? That's not what I wanted to do. Recommendation I have to get rid of. Okay. Lecture. Here. Okay. So here's the Exam 3 study guide. Hopefully you guys can see that.
Yep.
And yeah, when it comes to chapter 7, I'm more concerned with kind of the big picture as far as what are the main processes. So metabolism is all these chemical reactions that are happening in the cell. What role does catabolism have in that? And I want you to know more about the details of catabolism because it's important to understand that and how it's different in different types of bacteria. And that actually helps us, the understanding of the catabolism of the different types of bacteria helps us to understand which and identify which species is a pathogen, for example. We have a lot of tests looking at, does the pathogen grow aerobically? Is it fermenting? Is it able to ferment sugars like lactose or glucose and lactose or glucose but not lactose and things like that? And we can tell that from some of these products of the catabolic pathways. So while I want you to understand what anabolism is, you really don't need to understand any details of those processes other than the big picture and the definition. But you do have to know a little in more detail, not memorizing any pathways, not memorizing each step of glycolysis, but knowing what glycolysis is. What does it start with? What does it end with? What do bacteria or microbes that carry out respiration do after glycolysis to make more energy, energy being ATP, the energy currency of all life. And so understanding those kind of beginning and ending steps of glycolysis and the TCA cycle and what is the electron transport system doing for these microbes. And this may sound familiar because you've already learned about respiration in anatomy and physiology. And so bacteria similar to humans can carry out, some bacteria can carry out aerobic respiration. But note that it's different because bacteria don't have mitochondria, right? They're not eukaryotic in their cell structure, so they have no internal membranes where this is happening. Instead, that electron transport system in bacteria is on the cell membrane. And this, but at the same time, it's what you learned before as far as like the proton motive force being the force that is giving energy to convert ADP back into ATP, which is a really energy draining step. It takes a lot of power to get that phosphate back on so that you have ATP, adenosine triphosphate. So just looking at the lecture slides for, let's see, chapter, this is chapter 6, this is chapter 7, right? So just looking at the lecture slides for chapter 7, knowing those big picture processes, especially the importance of ATP as the energy currency of the cell, and then those other energy carrying molecules, NAD, NADP, or NADH in its electron carrying form, NADPH, FADH2. Those are also important in catabolism because they're able to carry the electrons from intermediate steps in the process of glycolysis or the TCA cycle, and then it gets shuttled into the electron transport system. And that's what gives the energy for in bacteria that carry out aerobic respiration, that gives the energy to make a lot of ATP. So just looking a little closer at this table from your book, you can see that for aerobic respiration, we get a whopping 38 ATP, theoretically, produced from one molecule of glucose, right? Whereas in anaerobic, it'll be a little bit less than that because the electron transport system doesn't shuttle as many protons out of the cell as it would with oxygen, which is like an ideal electron acceptor. It really grabs onto those electrons, and it's a good electron sink or an electron acceptor. But these other inorganic compounds, such as nitrates or sulfates, can also be important electron acceptors. They just don't allow for quite as much electrons to be transported through that system. So they get in the 30s probably, but not 38. And then fermentation has a dwindling amount of ATP that's produced by one glucose molecule compared to respiration because it doesn't produce any ATP at all through it's the only way it gets ATP is through the first step, which is glycolysis, that step that's common to both respiration and fermentation. And so it's just it actually ends up being a net of 2 ATP from glycolysis because 2 are used in some of the first steps, and four are generated. So four and then the two being used during the initial steps ends up being just two net ATP made by one glucose molecule going through a process of fermentation. So why do bacteria even bother with fermentation? It's so bad, such a poor energy yield. But the answer to that is that the other products are important uh, in enabling the bacteria to make more energy. So once you have an electron carrier, so if you look here at this, this is a figure in your book that is from figure 7.8 about fermentation. And what I just want you to take note of is these molecules highlighted in blue, which are the electron carriers uh, coming from glycolysis. So NADH is carrying an electron, right? Um, but it can't carry any more electrons unless it gets rid of the electrons that it's already carrying, right? So normally in respiration that would happen during the electron transport system, but in fermentation there's no electron transport system. So they actually get recycled through these variety of pathways that different bacteria or even yeast use. So you can see here, yeast converts that electron carrier back to its electron accepting form, right? The NAD plus in the process of producing ethanol. And then there are other products. There's lactic acid for some bacteria that are the E. coli makes a whole bunch of different products, including acids, alcohols, and gases. And actually, if I have a little another figure from a different source that's a little bit easier to understand because it's just simplified. You know, from pyruvate, which is the end product of glycolysis, what products occur from fermentation? Well, in E. coli you get different acids, alcohols, and gases in the form of CO2 or hydrogen gas. Um, Similarly, Clostridium has gases and alcohols and butyric acid. Enterobacter, so this is an important difference to notice. E. coli is an enteric bacteria, and so is Enterobacter. And if you're trying to figure out which pathogen is causing an infection, then you can look at the products of fermentation. Importantly, Enterobacter has 2,3-butanediol, and we set up tests in lab last week to measure if 2,3-butanediol is a product present in the metabolism of the bacteria that we tested. And that's the Voges-Proskauer test, right? So that would be positive for we were growing and testing Enterobacter, but it would be negative if we were growing and testing E. coli. Instead, E. coli would be positive for a mixed acid fermentation test, which is the methyl red test that we set up. So that you should just understand the products, because I want you to understand these tests that we're learning that are used routinely today in diagnostic labs to understand and identify bacteria and distinguish them from each other so that we can identify a pathogen. Yeah, other than that, you should just use the study guide to guide your learning in Chapter 7. You should know what reduction and oxidation is. And that's what's happening to the electron carriers, like NAD is being reduced when it's gaining electrons. Okay, so going through reduction to form NADH, and then oxidation to lose its electrons. Or you can think of the process of aerobic respiration is the full oxidation of glucose. So it's breaking down glucose, fully oxidizing it to CO2, right? So one glucose in the general reaction for glycolysis or for aerobic respiration is converted to 6 CO2 in the process. Um, and then also know the role of enzymes. I think I have that somewhere in here. Boop, 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 boop. Yeah, fermentation, energy carriers. Da, 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 da. Yeah, you should also know what enzymes do. So that's for chapter 7. But most of the material, as you can see, that'll be covered as chapter 6 and 13. Two of the open response questions are from chapter 6, the example of the gastrointestinal tract as a chemostat and the biofilm growth, and one is from chapter 13, the disinfectant question or topic. So I'll open it up again. Does anyone have any questions from any of the material, chapter 7 or other chapters?
I do have another, but I'll let everybody else have a turn first.
Okay, I think we might have lost Brian. Not sure if he was able to get sound. Paula, do you have any questions?
Yes, hi.
Hi.
My question is, how is the bacterial growth different from growth of eukaryotic microorganisms?
Oh, okay. So the way that bacteria grow is covered in chapter 6. And like all life, as you can see here in this table that I copied from another source, we already learned in chapter 4 about these elements that are required for organic molecules that are important for all life. And that's true of bacteria or eukaryotic microorganisms. It doesn't matter. But when it comes to reproduction, bacteria reproduce by a process called binary fission. Binary fission is a asexual process. It's basically a bacterium is making a clone of itself, right? So it's making, and here in the figure that I'm showing on the screen, one bacterium with a single circular double-stranded DNA genome, right, here at the top, must first replicate its genome. So it has to make an extra copy, so it has 2 copies of its genome. And once that happens, there's a new cell wall and a new cell membrane are formed by the process called septation, which actually makes physically one cell into 2 separate cells. But they're identical because they have the same DNA genome, the chromosome. Most often it's a single chromosome. So usually they show it just as one chromosome and it's a big circle like this. But when you look at it under the microscope, say an electron micrograph where you can see the detail of the genome, it's all kind of tangled up. So it doesn't really appear like in this cartoon a perfect circle. But if you stretched it out, it would be a continuous loop of DNA. So that's a chromosome. Extra chromosomal DNA is plasmid DNA that we learned about in chapter 5, I think. And that's just that extra piece of DNA that is a smaller circular unit of double-stranded DNA that carries kind of extra genes that if the bacteria loses that plasmid, it's no problem. It can still reproduce. But if it has it and it happens to be under conditions where the genes are beneficial, it'll give the bacterium a growth advantage. So the most common example, and the one that I want you to remember, is that a lot of bacterial resistance to antibiotics is carried on plasmid DNA. And those genes enable the bacteria to survive even if the drug is in their environment, in their growth area. Um, yeah, so that's the process that's prokaryotes that bacteria carry out. So how is it different from eukaryotes? Eukaryotes, remember, they have internal compartments, so their genome is not just in the cytoplasm, it's in the nucleus. So they have to go through the process. They can, for microorganisms, reproduce asexually or sexually, but they go through the process of mitosis, right? So where there has to be the disintegration of the membrane or the envelope, the nuclear envelope. The DNA is replicated, and then the chromosomes, which are usually several, line up, and the duplicate pairs line up and then separate. And so it's a lot more complicated because the cell has to be sure that it's getting all of one set of chromosomes to one side of the cell and all of the duplicate set to the other side, so that the microbe can be sure to, before it divides, be sure to have a complete set of that genome in the new cell, right? And then also you have other things that you need to worry about as far as the mitochondria some need to be partitioned into each cell and all that stuff. So mitosis is what I want you to remember as kind of the way that eukaryotes reproduce, which is very different from binary fission, which is fission, as the name implies, it's just splitting
Right.
into two.
Mm-hmm. Right.
Good question. Okay. Is that clear?
Yes, thank you. Mm-hmm.
Great. So this is the kind of growth that I'm talking about when I say vegetative bacteria, or then they're growing in this manner. When I say vegetative fungi, they're growing and dividing by mitosis, or they have other ways, like yeast will grow by budding, which is an asexual process but it still requires the disintegration of the nuclear envelope so that the DNA can be reproduced and then divided between the two cells. Yeah, so it's going to be a very different process.
Okay.
Any other questions about cell growth? I did go over some of this in class as far as the generation time or the doubling time, but I'd be happy to go over the calculation again if you need clarification on that.
Yes, we can do that.
Okay.
Mm-hmm.
So for bacteria, they don't grow, as you can see from the image we just looked at, which is a figure from your book. They don't grow in size. One cell creates two daughter cells of equal size, right? So when we're talking about bacterial growth, we're talking about growth or increase in the number of bacteria in a population. So remember when we did the cell phone experiment, or we swabbed our cheek and tried to grow bacteria from our mouth, each spot that we saw where bacteria grew came from one bacterium that came from our cell phone, for example, or one bacterium that was spread out from the many that we transferred using the swab, right? we separated them out using that streak plate isolation. So it was just one bacterium, but it grew to so many bacteria in that one spot that it became a colony, visible without the aid of a microscope. So that process is what we call bacterial growth, and that can be represented because every time there's a reproduction event, it's always a doubling. If you start with one, you have two cells. If you start with 100, after one reproduction phase, or it will have 200, there will be 200 cells in that population. And the way we can understand that is by this formula, n subscript 0 times 2, which is, it's always doubling, right? So it's that 2 is always fixed as far as it doesn't change. It's always 2 to the power of n, or as I mentioned in class, you can use the letter G to remember that that's the number of generations.
Right.
And N0 is the number of cells you start with in the, on the plate or in the culture tube or whatever you're trying to determine for the population. So if you start with one cell and it goes 3 generations, then it's 1 times 2 to the power of 3, which is 2 times 2 times 2, or 8. 8 times 1 is just 8 cells. And so, if I give you something like this on the exam, it will be very easy, something you can do in your head. You won't need a calculator. You just need to remember the formula, and I will provide you with the number of generations and the number of starting cells, starting bacteria.
All right.
Okay?
All right.
Yeah, and then the other thing that is important to remember is this bacterial growth curve, which in part 2 or phase 2, which is the log phase, that is the doubling. You can actually determine the doubling time from that, or so here we're calculating how many cells are in a population after a certain number of generations, but in this graph it can help us understand how fast the population is growing. And time for bacteria is often in minutes or hours because they double so quickly. E. coli under ideal conditions in the lab can double in just 30 minutes. So you can imagine how big the population can get if you're doubling every 30 minutes, right? And here, this is, the phase 2 is called log phase because it's the type of numbers that we're talking about are growing exponentially, right? 2 to the power of n. So that's an exponential growth because of the doubling. And so here actually, rather than having 1, 2, 3, or 100, 200, 300, we're actually going one full log order, so tenfold each number. So 10 to the 5th, right, would be 100,000, and then 10 to the 6th, a million, and then 10 to the 7th, 10 million. So that growth is really quick in the case of bacteria. Whereas in the human population, it's a linear because, usually one couple, so 2 people are having 1 or 2 or 3 children. They're not like doubling every time, right? We reproduce. So you can see that the numbers get really big really fast. But before they can get into that kind of ideal phase of where they're growing really rapidly and there's no limits on their growth as far as being the wrong temperature or not enough food and things like not enough water, things like that, they first have to prepare genetically, and that's the lag, L-A-G, lag phase, where they turn on genes maybe that are necessary for metabolizing the food, such as other sugars that they might need to be using or transporting those in that they wouldn't normally have turned on if they're not reproducing. So that in that case, the population or the number of bacteria don't change during that phase. So this is akin to, say, when bacteria enter the host body. So let's say you've eaten some contaminated meat, and that initial number of bacteria, they're not going to be able to grow. But once they get into the gut where they have plenty of food and maybe they can find space, they can do that step of attachment. Then they'll start turning on the genes so that they can multiply, and they'll start multiplying, right? So
Right.
it doesn't increase, the number of bacteria don't increase until those genes are turned on, and then once they have the proper temperature and they're in that environment where they can grow and have food, then they grow exponentially until something becomes limiting. Usually the food runs out. And then they reach the third phase, which is the stationary phase. And that's where there are still bacteria growing, but the number of bacteria that are continuing to divide is offset by the number of bacteria that are starting to die. So
Oh,
you
yes.
don't see, so they're still growing, but when you look at the whole population, it kind of flattens out here
Hmm.
because the number that are growing is equal to the number that are dying.
Right. Hmm.
And here is where like some of those extra genes may be turned on again. They're called secondary metabolites. So when we looked at our plates, for example, when we did the streak plate isolation, we had mixed three types of bacteria, and one of them had that red pigment where many of you noticed it was only in the middle of the colony, and around the edge of the colony it was still white. That white growth is similar to growth in the exponential phase. There's plenty of food at the very edge of the colony because that's the auger. It's touching the auger where there's new food around there. But in the middle of the colony where there's so many bacteria and now they're not near any, they've used up the food in that spot, they're not near food anymore, they transition to the stationary phase where some are dying and some are still growing. The ones that are still growing are maybe even using what's released from the dying cells to continue to grow since there's no food left on the auger in that spot. And then eventually the population will begin to die, and you'll see this gradual decline, not as steep as the growth part of the curve in phase two, but still will decline over time slowly. And that's the death phase, and that's because the number of dead cells is now kind of way over, much higher than the number of cells that are continuing to grow. All right. Any questions about these? Um,
I'm good.
Good. Okay. So back to the um, study guide, and I'm just going to check on the chat. So Brian has asked about mitosis and meiosis, which we already went over, and then is the temperature in the incubation connected with the growth curve? Okay, that's a good question. Um, the temperature, let's go back to where I was. Yeah, the temperature should be, in order to get the theoretically the best growth, you have to have the ideal temperature. And remember, for bacteria, all right,
Hmm.
there's a variety, they can pretty much grow anywhere depending on the type of bacteria, right? So here in this optimal temperature graph from your book, psychrophiles will be different species that are able to grow at low temperatures, say temperatures like inside your fridge. Mesophiles, though, are more around room temperature and body temperature, which is right around here where it peaks. Ideal temperature for mesophiles is body temperature, right? And then thermophiles can grow at much higher than body temperature rates, but they can't grow at body temperature. So they wouldn't be human pathogens that are in the group of bacteria that are thermophiles, because you can see down here around 40, the generations are 0 per hour, right? So they can't grow at that low temperature. And then hyperthermophiles have even higher ideal temperature that they grow at. So one thing that you can notice about these graphs is that they kind of gradually come up and then they dramatically drop at a temperature over the optimal temperature. So the optimal temperature being where this kind of peak, this rounded peak is in the case of each category of bacteria. So for example, a mesophile, if we have a fever, right, that raises our body temperature, that will really impact the ability of the bacteria to grow. And that's one of the great things about our body that helps us stay alive, is that the raising of the temperature by the fever can significantly slow down that growth. And if you raise it above this optimum, right, above this range, above 45, for example, there won't be any growth. And that's because the bacteria will die. Whereas lower, they might survive and kind of lay out, if you have contaminated food in your fridge, those mesophiles may be able to survive at a lower temperature so that when you eat them and they get in your warm body, then they can infect you. But if you cook your food, right, way up here around 90, 100, if you boil it, those things are dead and they are not going to be able to affect you. Okay?
Okay.
Any questions about temperature? All right.
No, I'm good with that.
So let me just check the chat again. I think I saw a question from... Okay. Yeah, is the drop in the temperature due to denaturing? It is due to denaturing. So this here, the drop in the ability to grow, mesophils, all of these different ranges, different groups, have special temperature requirements because of the enzymes that are important for their metabolism. And without enzymes, there is no life. So if you destroy the enzymes through heat, which irreversibly, remember heat denatures, denaturation is irreversible in the case of proteins, and enzymes are proteins. So they just won't fold properly, and if they can't fold properly, then that's it. They don't work anymore. And so that's true. If you introduce a mesophile, a bacterium like E. coli, which is a mesophile, it likes that body temperature best. If you bring it up above to, say, if it sits out in the hot sun, or if you boil it when you're cooking your food, then those proteins are denatured. It's like that example of the egg, the albumin in the egg white. It's all clear when you first open the egg, but then when you cook the egg, it turns white. That white egg is never going to revert back to that kind of clear, goopy form of the albumin. And when it's white, it's denatured. It can't work as it is intended to work. So indeed, the reasons that the bacteria can't grow anymore, especially when they go beyond this range, is because their proteins are dead, and if their proteins are dead, then the cell is dead. And these hyperthermophiles are like they have special enzymes that are able to function at the higher temperature, and so they work perfectly fine and grow perfectly fine at the higher temperature, and that's how their enzymes work. All right. I think that's it. Yep. Okay. So let's go back to the study guide. Any other questions on other topics?
I do.
Yes, Michael.
So, seems like from the questions and what we've covered, you know, we're not going to get. We don't need to know specifics about, certain media in terms of, I think we need to be able to discuss, the concept of, tests and stuff like that, but not to the level that we need to know,
Oh, yeah, don't.
macaque agar has bile salts and bile salts have this effect.
Yeah, no, you don't need to know that detail. I want you to know the basics of like temperature and pH, and you need to know detail of oxygen, really. Aerobes versus anaerobes, what enzymes they have that allow them to survive in oxygen, and what enzymes are lacking in those obligate anaerobes, and why they can't survive, why they're killed by the oxygen, which is converted into these reactive oxygen species at low levels. So if it builds up in the cell, it'll kill the cell because they'll be toxic. Um, Water avail, kind of all the basics, the basic elements, iron. Why do you need iron? Because that enzymes rely, some but not all, rely on iron to work. But what macromolecules have carbon in them? All of them do, carbon and hydrogen. What about oxygen? Where do you find that? What macromolecules need nitrogen? All of those nitrogenous bases, so the DNAs and the RNAs and also there's the amino acids that have that amino end, the NH2 at one end. So they all require nitrogen, but not carbohydrates and not lipids. So things like that, which is in this, it's kind of nicely summarized in this table that I have at the beginning of the lecture notes. But you don't need to know the tests that we're doing in lab in detail, but it will be relevant for that when you do your lab report on the, and when you're running the tests for the unknown project, it will become more relevant. So understanding this is important for understanding diagnostic testing.
Makes sense.
Yep.
Can you scroll to the right? There's no page number on that slide with the table.
Oh, the page number. This table is not from your book. It's just from the notes.
Oh, I know. So I want to know which slide it is that you're looking at in here. I
Oh,
can just scroll around and find it.
oh, it is, I guess the page is the second
Yeah.
page. Yeah,
Second page.
Okay.
this is the first page, and this is the second page of note. That's it's the, what's it called? Notes for chapter 6.
Okay.
Yep.
Thanks.
Yeah, remember siderophores are those we learned about in chapter 5 are those iron-binding proteins that are secreted by some bacteria, often pathogenic bacteria, that allows them to sequester iron. They also might have, like we talked about in lab, some ability to lyse, say, for example, red blood cells so that the iron is released, because the siderophores won't be able to really to bind the iron if it's still in the red blood cell. Let's see. Yeah, but you won't need to know the part of the that's related to lab that you do need to know is understanding what the difference between a pure and a mixed culture is.
Okay.
Remember we did the streak plate isolation, and that is kind of a precursor step to making a pure culture, because once you have individual colonies on the plate, then you can pick
From
one
them.
and be sure, because remember one spot, one colony on that plate arose from a single cell, right? So you know that that is one species,
Mm-hmm.
one clone. clonal population. And so by picking just that one spot, that one colony, and putting it in, say, a liquid culture of nutrient broth, then you're growing a pure culture. And the pure cultures, we need to have pure cultures so that when we do the diagnostic testing, the the results that we see we can attribute to just one type of bacteria,
Right.
presumably the pathogen, right? We have to separate out the pathogen from any of the normal microbiota. Otherwise, we'll never really be able to tell what the pathogen is because, maybe it doesn't ferment, but maybe one of our normal microbiota is a fermenter, and then we'll get a positive test result for the fermentation test, and then it'll just be way too confusing to tell what's what.
No, makes complete
Always,
sense.
yeah, pure culture. And then colony growth is different from biofilm growth, and biofilm growth is gone over in chapter 6 in a lot of detail. So other than knowing just those steps of biofilm growth, you should know um, kind of what's happening at the steps. So that this figure from your book is really helpful for learning the 4 basic steps that I want you to understand to form a mature biofilm, and then the 5th step is when it kind of breaks down and will go form biofilms at other sites. But there are more details than just these events that you should know, right? So the first is that there's kind of reversible attachment, and only biofilm forming species can do this first step. Not any bacteria can do it. And then once a monolayer or the bacteria begin to multiply in a single layer on the surface, they will communicate with each other through quorum sensing, and that quorum sensing will help them to signal to each other that, okay, we're going to irreversibly attach and start to grow in these, start to produce exopolysaccharide and grow in this three-dimensional kind of upward tower. And the polysaccharides that are secreted from these cells are kind of a sticky glue that has many functions. It is important for kind of holding these towers together. It prevents the entry of things like antibiotic drugs or immune cells. And so it's really an important part of the biofilm, is this kind of gooey slime that covers everything as far as the polysaccharides that are, they're exopolysaccharides because they're secreted from the cells and they become this kind of slimy matrix that the cells are able to grow in. And other cells that are not biofilm formers are able to join into that community. But it's really tough to, because of the three-dimensional aspect of the growth and because of this protective exopolysaccharide-rich coating over the population that's growing. It's really hard to treat these and cure biofilm infections, and that's the medically relevant part that I want you to remember, is that these are chronic infections. When one biofilm, if the nutrition is no longer available in that site, it can just break down and form another biofilm somewhere else. But it can't form biofilms anywhere. Biofilms are best formed on surfaces that are non-shedding. They're smooth and non-shedding. So the most obvious natural biofilm that we have are on our teeth. But biofilms can form in some parts of our body, but especially if we have some artificial device within our body. So like a catheter or an artificial joint, that's where these biofilm infections can occur and become very chronic and serious and life-threatening. And then I realized that I didn't go over the oxygen requirements, so if anyone wants me to go over that, I'd be happy to do that as well.
Yes, please.
So the oxygen requirements, there's a figure in your book that kind of lumps it all into one, but I actually like this figure that I've taken from a text we used to use better because it shows you individually how each type of bacteria um, grows, whether it's in the presence of oxygen as far as obligate aerobes. So aerobes require air or oxygen. Anaerobes don't require it, but some can use it. So if they're obligate, that means they have to grow in the presence of oxygen. They only can carry out aerobic respiration as their metabolism, and because of that, they need that electron acceptor. They need to use oxygen as an electron acceptor. If they don't have oxygen, then uh, they won't grow. So you can see how they grow just at the top of the agar in this tube because the air is there, right? There's no air at the bottom. The air can't diffuse through the solid agar. On the other hand, on the opposite spectrum are the obligate anaerobes. They will die in the presence of oxygen. Why will they? That's because they lack these enzymes like superoxide dismutase or catalase that break down the toxic reactive oxygen species like superoxide that or hydrogen peroxide that will form as a result of oxygen just naturally being anywhere. It kind of at a low level converts. But if those toxic species accumulate in the cell, then they are very reactive and they'll disrupt the function of the proteins. And remember, the proteins include enzymes that are necessary for the cells to keep alive, because if they can't have these enzymes catalyzing their reactions, then they can't survive. And that's the case for the obligate anaerobes. So they don't use oxygen they don't carry out aerobic respiration. They can carry out anaerobic respiration or fermentation, but they don't use oxygen, and they actually are killed by the presence of oxygen. So no growth at the top, only at the bottom. Facultative anaerobes are these kind of very flexible bacteria, things like E. coli, that they can grow in the presence of oxygen. They can use oxygen to grow very efficiently. You can see it's a lot of bacteria growing here at the top where the oxygen is the richest, but they can also grow by fermentation, which allows them to grow where there's little or no oxygen present. And then there's some special cases with the aerotolerant or the microaerophiles. But these three are the big ones. Any questions about that? Let me see. These are actually, so Brian was asking, are these anaerobic, uh, are these blood tubes? And they aren't blood, they're agar, and they would just be a pure culture of bacteria grown in here. So you still, even with these tests, you have to have a pure culture of bacteria. Otherwise, you might have a mix of obligate aerobes with facultative anaerobes, and you can't physically see the difference, so that you would just see growth all the way through, and then it would be confusing what trait is associated with the pathogen. So these are only one type of bacteria in each tube, one species. Any other questions? And the pH and the temperature would be ideal, like just put it in the incubator like we put the plates in the incubator. And the agar itself is not a nutrient, but it will contain other compounds in the agar that are things like glucose. If it's a defined media, then we know exactly what's in it. But most often we use things that are complex media in the lab, and so we don't know exactly what's in the ingredients that's allowing the growth, but we put, a certain amount of yeast extract. So there's probably not everything in yeast extract is important for the growth, but some things are. And so we just add the yeast extract because we know that's supporting the growth. Okay, so I haven't gone over much of, and it's just 1 o'clock now, but I haven't gone over much of chapter 13. So let's look at that for a minute. The first, so remember that I want you to know in detail the story of alcohols and how chemical disinfection is heavily dependent on the type of the microbe that you're trying to control. So we learned about growth in chapter 6, and now in chapter 13 we're learning about how to control the growth of microbes. So we learned about lots of different kinds of microbes, not just bacteria, but in general, as I mentioned in lab, you can divide them into kind of two groups as far as being those microbes that are resistant to control, and that's chemical control, or physical control like heat, and those that are susceptible. So generally, vegetatively growing microbes, whether they're bacteria or protozoa or fungi, they're more susceptible. They're easier to control, easier to kill with chemicals, easier to destroy with heat. So, for example, that being a physical means of control. But when these microbes form dormant or protective forms, such as the cysts of protozoa, endospores of bacteria, those forms, or if they just inherently have something about their structure that allows them to be more resistant. So the waxy outer coat you can think of as, when you wax your car, it does a really good job. Like when it rains, the water doesn't penetrate. It just kind of balls up and sits on that surface. And that kind of waxiness, that kind of property is true of the mycolic acids that surround the Mycobacterium tuberculosis and other species of the genus Mycobacterium. And so they are more resistant to physical, like heat control, and chemical, because the chemicals don't penetrate very easily. Same thing with endospores, and prions are these infectious proteins that are really wacky because unlike most proteins, which can be easily denatured by heat, when I talked about albumins in eggs turning white when you cook your egg, those kind of things, they're usually very sensitive to heat, but not prions. Prions are heat-resistant forms of an infectious protein and really tough to control. So how do we control them? Well, the way we do in the lab is with the combination of high heat that results in steam at 121 degrees plus pressure. And within 20 minutes you can get rid of all the microbes on that list, including endospores, except for prions. Prions require a higher temperature, 132 degrees, for 4.5 hours. Okay, so let's see, do I have a picture of, in your book there's a picture of the, oh here it is, how the autoclave works with steam, and there's a chamber that creates the high pressure that really allows that heat, the steam allows that heat to penetrate. And the example I gave when I talked about it previously was like, think of your pressure cooker. You cook your food much faster in your pressure cooker than in the dry heat of the oven, right? It takes hours in the oven, but in minutes in the pressure cooker. So it really, once that heat penetrates the material, it's very effective at controlling the growth and destroying really all of the structure associated with that microbe. But let's look at some of the chemicals that are used to control um, growth, and they come in a variety of um, levels of effectiveness that are divided by the CDC into three basic levels. High level disinfectants, and this is table 13.2 in your book, mid-level or intermediate level, and low level. And really the only difference is that the high level and the intermediate level are taking care of destroying these really tough microbes that are resistant to control. Things like mycobacteria and endospores. Whereas like vegetative bacterial cells, vegetative fungal cells, most viruses are really easily controlled by even low-level disinfectants like alcohols. So let's take a moment to look at alcohols, because remember when we did the gram staining, we were actually using alcohol to see that difference between the gram-negative cell wall and the gram-positive cell wall. Really the gram-negative cell envelope, because it's that outer membrane of the gram-negatives that's destroyed by the alcohols. So in that decolorizing step. And that's true of anything that's surrounded by a plasma membrane, so that lipid bilayers are very sensitive to alcohols. So the outermost layer in gram-positive, in gram-negative bacteria is going to be destroyed by the alcohols. And even the inner cell membrane that's covered by the cell wall of in the gram-positive, the many layers of peptidoglycan in the cell wall of the gram-positive, if you leave it long enough in the alcohol, then it will be destroyed as well, because the cell wall is just fully permeable. That alcohol gets right in past the cell wall to the cell membrane and destroys it. And that's true of some viruses have a lipid bilayer. They're called enveloped viruses. They have a lipid bilayer, and they are easily killed by and destroyed by alcohols. So that's the plus. But the minus here is the non-enveloped viruses, which don't have a lipid bilayer at all. They have no lipid content. Instead, they're covered by a protein shell called a capsid. And all viruses have a capsid, but some have this outer layer of the envelope that covers that capsid. So the capsid is not as easily destroyed. It's not destroyed by the alcohols. These low-level disinfectants don't destroy it. So that's where things like, hand sanitizer comes in as being kind of protective in some cases, but not in all cases. And that's because viruses that are not enveloped, like norovirus, which is a very contagious, basically stomach bug that causes severe diarrhea, nausea, and vomiting. And whereas other viruses like the flu virus, which has an envelope around it, the common cold virus, those are destroyed easily by the alcohols present in hand sanitizer. The other thing I want you to understand about alcohol is that the way it works is it has to, in order to penetrate the microbe, water has to be present. So if you were to immerse these bacteria or viruses in 100% ethanol for the bacteria, and then try to grow them, like we did in the disinfectant lab, where we waited a certain amount of time, and then after the bacteria were sitting and then disinfected for 2 or 5 or 10 or 15 minutes, and in this case they're just doing seconds, not even 1 minute. We took it out and tried to grow it, and if you put it in 100% ethanol and you took it out after 10 seconds, or even after almost a minute, 50 seconds, it would grow no matter what. But going down and mixing it with a little bit of water and getting down to 95%, it's very effective at penetrating the membrane and destroying it. And so there's no growth in this optimal range of about 70 to 95, even at 60%. But usually hand sanitizers are somewhere, if they're going to be effective, they're somewhere between 70 and 95%. Below that, you get kind of, it depends on time. Like if you use the hand sanitizer more than 10 seconds, then, up to 30 seconds, then yeah, probably it would protect you. But otherwise, it's not going to be effective lower than 50%. Okay, so I'm just going to finish up because it's already 10 after, so I'll just open up to one last question. Anyone have one last question that they would like to? Alcohol versus hydrogen peroxide. Okay, so, oh, and antimicrobial versus, okay, I missed a few good questions from Brian. Sorry, Brian. Prion is not dormant. It's not like a spore. It has one form. It's the diseased form, or it has two forms. The natural form that's healthy in our bodies and the diseased form. And if even one molecule of the diseased form is present in our nervous system, it will convert all of the healthy form to the diseased form. That's the only forms it has. It's a protein. It can't become a spore because a spore has these many layers and it's all dependent on gene expression. But being an infectious protein, there are no genes. So it's just a protein, and it's just a matter of it misfolded, and it still worked, but it worked in a detrimental way in that misfolded form.
Before you move on,
Sorry?
before you move on, since you're on
Oh,
prions,
yeah. Yes.
just to clarify, a prion in its non-folded form is just a regular functioning protein. So if
A
we
regular,
say,
healthy, functioning protein, yes.
and then it misfolds, and then that's really when it becomes a prion. Before, otherwise, it's really not one, right?
Yeah, it's, they call it a diseased form, or, yeah, scrapey protein, or there are other names for it. Yeah.
Okay,
Yeah, but
good. Thank you.
prion is the diseased, the infectious form. Yeah. Yeah. Whereas if you eat contaminated meat that has like bovine prion in it, you will get mad cow disease. Over a period of time, it's very slowly progressing disease, but eventually you will get a fatal case of mad cow disease. Yeah, and they don't know before. You just have these kind of dementia, kind of neurological symptoms, and then once the autopsy is done, they can see and diagnose whether you had prion disease based on the tissue from the brain. Yep. Yeah, and then does that clarify that for you, Mike?
Yes, thank you.
Okay. The difference between antimicrobial and antibacterial. Antimicrobial is referring to any microbe. Antibacterial, it's only bacteria. Okay? Antimicrobial is a more general term that includes bacteria. And then pH level. The pH level can inhibit growth. It can sometimes kill. So, but it's not alone enough to act as like a disinfectant. You couldn't just use, um, like have you ever put like lemon juice on your meat when you're marinating it? That acts that lowers the pH and acts to kind of keep the microbes from multiplying more and contaminating that meat, right? So pH
So
can
you can do it
help,
with an
but
apple to prevent drowning.
it doesn't like destroy them. So it doesn't. So it's not a. It's like a very low level acids are a very low level disinfectant. And then alcohol versus hydrogen peroxide. Hydrogen peroxide. Well, alcohol. They're both Hydrogen peroxide can be a high-level, actually, disinfectant. I think I went over it, and your book certainly goes over it. Um, they are able to, in the gaseous form, really destroy proteins, just like ethylene oxide can. So also peroxides.
Mm-hmm.
So they come under peroxides, I think, are under chlorines, which are pretty fair at uh, controlling both endospores and mycobacterium. But really, no chemical is effective, reliably effective at destroying prions. So you can use this gaseous form of ethylene oxide to really sterilize things, but it won't apply to prions. But prions aren't just kind of floating around in the environment. They're in special sites that you could then, once you know that they're there, you can deal with them appropriately. Um, any other questions before I close?
Not for me, but thanks for doing this.
Thank you so much.
Okay.
Yeah.
All right. Don't forget to review the positive gram-negative cell wall differences from Chapter 5. That's something you really still need to know. Okay. Thank you
Okay.
for
Thank,
joining, and good luck.
thank you
Thanks
so much.
again.
Okay.
See you tomorrow.
Bye.